Hi everyone, and welcome to Chapter 7, which is on cellular respiration. And I will give you all fair notice that Chapter 7 and Chapter 8, which are the cellular respiration and photosynthesis chapters, are usually everyone's least favorite chapters. So I'll try and make this as painless as possible because I know that these are chapters that aren't as popular with the students. Now, when we talk about cellular respiration, the first thing you have to remind yourself is that this process is for acquiring energy. It's for cells to get energy. And if you think about it, where do they get energy? Well, where do you get energy? From breaking down food, right? So in the case of cells, they acquire energy from breaking down carbohydrates specifically. Okay, so remember in previous chapters we referred to carbohydrates and sugars as quick energy. Well, it's the same thing for a cell. So they break down carbohydrates that have been produced by photosynthesizers, which will be in the next chapter that we talk about. And that's why the two processes, photosynthesis and cellular respiration, are opposites. Now, what they do in cellular respiration is just like when you have respiration, you have breathing, they take in oxygen and release carbon dioxide. Now, as soon as you hear that they're taking in oxygen, that tells you that it's an aerobic process. So you can circle aerobic and remind yourself that that means requires oxygen. Okay, so aerobes are cells that need oxygen. Now, when I say what does that remind you of, it's what I just mentioned a second ago, which is your own breathing. Okay, when you have breathing and respiration, you breathe in oxygen and you release carbon dioxide. And then plants, big trees, they use that carbon dioxide to make their food and ultimately energy in photosynthesis. Okay. Now, when we say that in cellular respiration you are breaking down carbohydrates, specifically we mean glucose. Okay, so we have that right here. And you can see that down here in the cellular respiration formula where you see glucose, which is the carbohydrate, gets broken down. So it's losing electrons. So Leo says Ger, right? Leo lose electrons, oxidize or oxidation. And then through this process, you get energy being created. Now, see, in this cellular respiration, the breakdown of glucose into carbon dioxide and water, it brings back that idea of redox. So like I just pointed out, oxidation is the first side of things. Remember, the carbon of glucose getting oxidized. And then the opposite is happening with the oxygen that the cell is taking in. The oxygen is getting reduced, and remember I said it's funny that we say reduced because look, it looks bigger, doesn't it? But it's reduced because it gained those negative electrons, which you see here as the H2. Okay, so just always, if you remind yourself of redox and that little trick of Leo says Ger, then right away you'll know losing electrons is oxidized, gaining electrons is reduced, and if you're visually looking at the formula, the one that suddenly looks bigger is actually reduced, because remember we said it's weird like that. Now, when you think about any process in a cell, you always want to remind yourself where it happens. So in the case of cellular respiration, always remember that it takes place in the mitochondria, and that's why we call the mitochondria the powerhouse of the cell, because it is where the making of that energy is going to happen. And when we say making of energy, keep in mind, we're talking about getting ATP. And in a few slides, we'll talk about exactly how this ATP is being synthesized. But notice you have ADP, so that's diphosphate, meaning two phosphates, gaining a third phosphate, so now it's triphosphate. And what did we say in previous slides about ATP and its energy? that energy for the cell is based on those 3 phosphate groups being linked, because you can then break that. Okay, so whenever we learn new concepts, just remember to connect it to what you've learned in the past so that you're really solidifying these concepts. Now, when we talk about cellular respiration, there are 4 phases. And what I want you to know when we talk about cellular respiration, and even when we go into photosynthesis as well, for cellular respiration, make sure you know the names of the 4 phases, which are glycolysis, prep reaction, citric acid cycle, which is sometimes called the Krebs, K-R-E-B, Krebs cycle, and the electron transport chain, which you'll also see the word chemiosmosis associated with it. So that's right here. You may want to circle that word chemiosmosis, because that will come up as being important later. Now, in addition to the names of the phases, I also want you to know whether each phase requires oxygen or does not. And how do you know if it requires oxygen or not? Remember the term aerobic. So aerobic, circle that term, and write requires oxygen. Anaerobic does not need oxygen. So anaerobic means this phase, glycolysis, can occur without oxygen present. Okay, it's the only phase that can occur without oxygen present. All the other 3 phases, they say aerobic. And if you want a little trick to remember that aerobic means with oxygen, think of air. Air for you is your oxygen source. So aerobic means it needs oxygen, air to occur, whereas anaerobic does not. The third thing I want you to know for each of these steps is how much ATP, how much energy will a cell get from that particular phase or that particular step. So notice you have 4 steps, 4 phases. The first one, glycolysis, gives the cell 2 ATP. And a cell can get that 2 ATP whether oxygen's present or not, because that's anaerobic. It doesn't require oxygen. So anytime a cell is going to break down glucose, whether oxygen is present or not, it will get at least 2 ATP from that glucose. Also make note that this is the only step that is occurring outside of the mitochondria. So glycolysis is actually before the main cellular respiration steps, and it occurs in the cytosol, that fluid of the cell, not in the mitochondria, whereas the rest of the steps occur in the mitochondria. Now notice again, the rest of the steps require oxygen, and when I show you the prep reaction, notice I highlighted in red prep. When you look at this slide, I want you to ask yourself, how does the prep reaction differ from the other 3 steps of cellular respiration? What do you notice about it? All of the other steps have red highlighted ATP, right? Prep does not have any ATP produced. Okay? And the trick to remembering that the prep reaction is different from the other steps in that it does not produce ATP, it doesn't give the cell any ATP, is the idea of prep. Okay? Think about yourself. If you're cooking a meal, right? You're going to have some chicken and mixed vegetables that you're going to saute up. Well, what do you do first? You usually cut up that chicken, right? You cut it up into, usable sizes. But you can't eat that chicken right at that moment and get your meal and get your nutrients right there just by cutting it up, right? You then have to cook it. But you cut it up to prep it for cooking it, for making the meal. You're preparing it, okay? Now, technically, you could eat it, but that's a whole other story. That's for when I teach microbiology. But when you do any kind of prep, you're not getting the end product yet. So in the case of cellular respiration, when you do the prep reaction, you're not getting the goal of ATP yet. Instead, what you're doing is you are breaking down the substances into usable substances for the next step or the next cycle or next phase of cellular respiration to then make ATP. Okay, so the prep reaction, you get no ATP, but it breaks down the substances to then go into the citric acid cycle. And in the citric acid cycle, then the cell is able to get to ATP. Okay, I am not going to bother you about the actual intermediate products, so things like acetyl-CoA, things like NADH and FADH2. I won't bother you with those because this is not biochemistry class. For your purposes, it's more important to be familiar with the steps, whether they're requiring oxygen or not, and what are they giving that cell in terms of energy. So citric acid cycle, you see, okay, 2 ATP. A little bit of energy. That's better than the prep reaction gave, right? And then you hit the electron transport chain. And what do you notice about the electron transport chain versus all the other steps? Boom! That's where you get the bulk of ATP. And that's thanks to chemiosmosis, which I told you to circle before, that we'll talk about in later slides. So the electron transport chain is where the cell will get the most ATP. Now, when you take all of this at once and you ask yourself how much energy, how much ATP is the cell getting from breaking down one glucose, because this was all breaking down a single glucose molecule, well, it could get up to 2, 4, plus 34, up to 38 ATP it could get out of cellular respiration. But with cells like ours that are eukaryotes, they then lose 2 ATP to move that energy out of the mitochondria where it's just been produced, because the mitochondria, remember, is a membrane-bound organelle. You made the ATP in here, but now you want to boot it out and get it into the rest of the cell. So that's kind of like paying a toll to get it out. You got to use some energy to get it out to the rest of the cell to use it up. So then that would give 36 ATP. Now, I know that's a lot at once, but we're going to go through each of the steps one by one. But first, I want to show you a visual just to kind of help you see what's going on in this process. So here you have the mitochondria, okay? And here you can see the different processes occurring for cellular respiration. Again, notice, where's glycolysis? It's outside of the mitochondria. So glycolysis happens first outside in the cytoplasm. And what did we say it yields? It yields 2 ATP. So you can circle that 2 ATP to remind you from the glycolysis step. Again, this step can happen without oxygen. So a cell does not need oxygen for this to happen. Then you get to the prep reaction, and what do you notice? No ATP, but it is now occurring in the mitochondria, and it helps break things down to go into the citric acid cycle where the cell can get 2 ATP. Then those products go into the electron transport chain and chemiosmosis, and that is where you get the bulk of ATP produced. And the cell can make up to 34 ATP in that electron transport chain. So the maximum you could get from one glucose is 38 ATP. Now going step by step, first up we have glycolysis, and again, I know some of this will be repetitive, but repeating and repetition helps you to really remember the significant points. So again, glycolysis, where did we say that occurs? Outside of the mitochondria, in the cytoplasm. And what did we say is the significance about this step compared to the rest of cellular respiration? Well, as soon as you see glycolysis, I said remind yourself that it can occur without oxygen. Okay, so whether oxygen's present or not, a cell can break down glucose in glycolysis and get to ATP. because it is anaerobic. Now, in order to get the ATP, there are two steps of glycolysis. There's the energy investment step, in which 2 ATP are used to split a glucose molecule in half, and then the energy harvesting step. Okay, so it took some energy to break that glucose molecule, but now from those halves of glucose, the cell is going to be able to yield 2 ATP. And it does this with what we call substrate level phosphorylation, which I'm going to show you a picture in a minute. I just first want to show you a summary, a visual summary of the inputs and outputs of glycolysis. So glycolysis, we said, you are taking one glucose molecule and you are starting to break it down, cut it in half, and start to get ATP out of it. Now we said that in the first step you have to break down that glucose, which takes some energy. So that's why you see that ultimately you use 2 ATP to break that molecule, and then you end up building 4 ATP. So whenever we say you get 2 ATP out of glycolysis, it's what's called a net gain. So the cell had to use or put into ATP to make 4 ATP. So in that process, it gained a total of 2 ATP. Okay. Again, I won't bother you about the 2 versus 4. I just care that you know glycolysis gives the cell 2 ATP, and it gives it whether oxygen is present or not. And the way that it gives that ATP, or the way that it makes it, is through what's called substrate level ATP synthesis. You will see this come up a lot in this class, so circle star highlight that term, substrate level ATP synthesis, and remind yourself that, again, the key to ATP being a high energy molecule lies in the phosphate bonds. The fact that it has 3 phosphate bonds, and that you can then break those to get energy. So substrate level ATP synthesis is building those phosphate bonds, because that's what you need to later on use for energy. So what it means, or what it does, is there's an enzyme that uses a substrate to provide a phosphate group to what's called ADP. And remember earlier I said ADP stands for adenosine diphosphate, meaning 2 phosphate groups. So substrate level ATP synthesis uses an enzyme as a little seat to help get a phosphate group from a substrate, which in this case is PEP. And it gets that phosphate group, adds it to ADP, so now you have ATP for triphosphate, 1, 2, 3 phosphate groups. And that's where the energy of ATP lies. So now that the cell has built this bond, right, it could then put that ATP somewhere else in the cell to break that bond and get energy when it needs it. Okay, so that is substrate level ATP synthesis. You will see that come up a few times. So that's getting a phosphate group from a substrate, adding it to ADP to make ATP. Now this is just a big figure to remind you, or to summarize glycolysis for you. Again, it shows that there's energy investment and then energy harvesting. I want to emphasize the fact that you are splitting a single glucose in half. Okay, so up here you start with a single glucose. You're splitting a single glucose in half and then going through a series of intermediates to get to the key step, which is ATP synthesis. So down here you get to substrate level ATP synthesis. Remind yourself what that means. And then at the end, you get a yield of 2 ATP. Okay? And if you want, you could put a little star next to the figure and write anaerobic. No oxygen needed. Okay, just to keep reminding yourself that anytime you see glycolysis. Now, throughout these slides, you may have noticed that during glycolysis, the end product that was listed was called pyruvate. Okay, now pyruvate is an important molecule because if oxygen is present in the cell, okay, that pyruvate will then enter the mitochondria and go through the rest of those stages of respiration. So it will enter the mitochondria and it will go through the prep phase, it will go through the Krebs cycle, and it will go through the electron transport chain. And ultimately, you can have that cell get between 36 or 38 ATP out of glucose once the pyruvate has gone through all of those steps. Now, if oxygen is not present, if there is no oxygen present, instead, the pyruvate will undergo what's called fermentation. Okay, fermentation occurs in the cytoplasm, and ultimately, it does not produce any extra ATP. And we'll talk about that in a minute. And you can see here, if the pyruvate goes the no oxygen route and goes through fermentation, you can circle this part of the figure here and write, this is fermentation. Fermentation, at the end, you get lactate and ethanol. So sometimes it's written as lactic acid and ethanol. Okay, and we'll go through now, why would a cell want to do that? So with fermentation, I just said, okay, you're not going to get any extra ATP. The cell, however, can do this in an anaerobic environment, meaning no oxygen is present. It does not need oxygen. So if there's no oxygen present, what a cell will do with glucose is it will first go through glycolysis, because remember, we said that's also anaerobic. So it will put glucose through glycolysis and get to ATP. It will then move it into the fermentation stage where it further breaks down the pyruvate, in this case to lactic acid or alcohol. Okay, you've probably heard it as lactic acid fermentation or alcohol fermentation. And ultimately, this is still useful to the cell because even though it does not produce any extra ATP, it does break down the pyruvate into intermediates that can then be used for glycolysis again. And what do you know about glycolysis? Well, glycolysis, the cell can get to ATP, and that's better than nothing. So the cell can then cycle through glycolysis and fermentation until oxygen is present. So if I asked you about, what happens to a glucose molecule if there's no oxygen? How much ATP can a cell get from glucose if there's no oxygen? The answer is it goes through glycolysis and fermentation and the cell gets to ATP. If, however, oxygen is present, then you would tell me that glucose goes through glycolysis, prep, citric acid cycle, and electron transport chain, and that you could end up getting a maximum of 38 ATP produced, okay, or net gain. Now, when we talk about fermentation, I've kind of already mentioned the efficiency of it, but this slide I put together to kind of summarize the pros and cons of fermentation for you. Why would a cell want to do this? Well, in the case of when there's no oxygen present, it still provides a quick burst of energy, such as for muscles, and that quick burst of energy is at the efficiency of 2 ATP per glucose that it breaks down. And again, that ATP is due to glycolysis occurring as part of the fermentation process. Now, the bad part is that at the end of this process, we said that you end up having lactic acid or alcohol, and these can be problematic to cells, okay? They can change the pH and they can put a strain on oxygen to try and get rid of this kind of toxicity that is now present. Okay, so a cell wouldn't want too much fermentation to keep happening. There's kind of a limit. And if you think about muscle fatigue, then if you've ever, worked out too much, you know what happens as a result of these toxic byproducts building up. Okay, but again, it still has its value. It still gives the cell 2 ATP, which is better than no ATP. Now, what about if there is oxygen? What happens next after glycolysis? Well, instead of fermentation happening, instead the pyruvate will go into the next step of cellular respiration. And that step is the prep reaction. And again, we've already mentioned that this occurs inside the mitochondrial matrix. And what's something important that you have to remember about the prep reaction versus all the other reactions? No ATP is produced. Instead, the purpose of this reaction is to prepare the molecules for the citric acid cycle that comes next. And the way it prepares it is you get conversion to what's called acetyl-CoA. But again, like I said, I'm not going to bother you about intermediates such as acetyl-CoA or things like NADH. I focus more on the reaction, when it's occurring, is there oxygen or not? Where is it occurring? And what does it give the cell in terms of energy? Now, like I said, the prep reaction has helped prepare the pyruvate into an intermediate that can go into what's called the citric acid cycle. And remember, the citric acid cycle now will produce 2 ATP. So I want you to circle this output here of 2 ATP. Okay? The way that this occurs is through substrate level ATP synthesis. And again, remind yourself what we keep saying. The key to ATP lies in the phosphate bonds. Okay, that's what gives it its high energy aspect. And so to build ATP, which is ATP synthesis, the cell puts together those phosphate bonds. And then when it needs energy later on, it breaks the phosphate bonds. So again, substrate level ATP synthesis is when a phosphate is transferred to ADP to form ATP with the help of an enzyme. Once that has occurred, then the next step and the final step of cellular respiration is the electron transport chain. And like I said, the electron transport chain is where the cell will get the bulk, the biggest amount of ATP formed. Now remind yourself that this is aerobic and it requires oxygen. And the reason that is that oxygen will be the final electron acceptor in this electron transport chain. Now again, this is occurring in the mitochondria. And in the case of prokaryotes, instead it would occur in the plasma membrane, because remember, prokaryotes don't have internal membranes, internal organelles. Okay. Now, what the electron transport chain is a series of carrier molecules that are passing electrons from high energy state to low energy state. Because again, think in nature, in balance, things want to go from high to low. That even works with energy. So electrons are passed from high energy states to low energy states. It's basically a complex mixture of proteins and what's called cytochromes, which are proteins that are tightly bound to iron atoms. Okay. Now, again, that's something that I won't bother you about, the idea of proteins and cytochromes. I just want you to know that the flow of the electron transport chain is moving electrons from high energy to low energy, and then the cell gets that energy throughout each step. Okay, because you're going to lower energy, so that high energy has to go somewhere, right? The cell is able to harvest it. Now, at the end, it's oxygen that serves as the final electron acceptor, and we also in this process have coenzymes. And what coenzymes again mean, these are things that are inorganic, so metals or minerals, that help enzymes function. And a lot of what's going on in here is, enzyme based to produce that energy, because like we mentioned before, a lot of times ATP is produced by substrate, right? That ATP synthesis, substrate level ATP synthesis, which is what you see here. You have these ATP synthase complexes. And so what this figure is to show you the idea of the mitochondria, and you have the electrons being passed along, and it's being passed along a series of carrier proteins. So each of these big blobs of orange are carrier proteins. And as they're passing the electrons along, the electrons, you then see the idea of having hydrogen ions here. You get those going through a channel that's called ATP synthase, and that is producing ATP by that substrate level ATP synthesis that we talked about before, the idea of having ADP get an extra phosphate bond to produce ATP. That ATP then leaves the mitochondria in order to be used throughout the cell. And when it's used throughout the cell, what did we say? ATP is a high energy molecule because it has those 3 phosphate groups. So when you get that ATP to wherever in the cell needs energy at that time, that part of the cell will then break one of those molecules, one of those phosphate bonds as energy, right? So what do you have now? You now have ADP. What's that ADP going to do? Go into ATP synthase. So we have that here again, and get a phosphate again, so that now it can be ATP to leave to get used by the cell again. Okay, so you have this constant cycling in cells between ADP, which has potential to be energy, and ATP, which actually gets used as energy. Okay, like I said, this is not biochem, so that's why I'm not going into the details of NADH and FADH and all of that. That's not significant for this course. Okay, if you are interested in learning more about that, then contact me and Remind. I can send you some videos that usually help out students, but again, that's beyond what you need for this lesson and this course. Okay, instead focus on the fact that the electron transport chain is where you're getting the bulk of ATP. You notice that here with ATP synthase complex, it shows chemiosmosis. Okay, and chemiosmosis is linked to this hydrogen gradient, this ion gradient feeding in the ATP synthesis to help produce all of that ATP. So again, make sure you know electron transport chain is where you hear the term chemiosmosis, and that you have the most ATP being produced during this step through ADP gaining a phosphate. Okay, that's important to keep remembering the idea of ATP versus ADP. Now this figure here is to show you the whole process of getting any kind of ATP out of this one glucose. So the whole process of cellular respiration, again, it starts off with glycolysis, and notice this is in a different color because what did we say about glycolysis? 2 significant things that distinguish it from the other steps is it occurs in the cytoplasm, so circle cytoplasm for glycolysis, instead of the mitochondria, and it does not require oxygen, so no need for oxygen in this step. And then the other significance we said is it produces 2 ATP. If oxygen is present, then that broken up glucose, which produced 2 pyruvates, those pyruvates will now go into the citric acid cycle. And in the citric acid cycle, you get another 2 ATP produced. Now remember, now at this point, we are in the mitochondria and oxygen is required. Then the products will move to the electron transport chain, okay? And that's where the rest of the ATP is produced, okay? So you get this net yield. Now notice that this slide was focusing on the overall energy. So what did we leave out here? The prep reaction, because you're not getting any energy from that. Okay, so if you want to remind yourself, you can squeeze in that the prep reaction would be occurring over here before the citric acid cycle, but that they left it out from this figure because no energy, no ATP is produced in that process. Okay. Now, if you have any questions or concerns on all of that, because I know it is a lot to take in, but again, remember, it's repetitive. So if you focus on the repeat parts, the significant parts, it's not that bad. If I didn't mention things, so for instance, the conversion of NADH and FADH to NAD to FAD, then that means you don't have to know it for our exams, for our course. Okay, so you'll sometimes see extra information in the textbook that we don't cover in the lesson because it's extraneous. It's not significant for our purposes. Okay, but again, if you want me to review anything or give you practice problems on cellular respiration versus fermentation, anything, just contact me and remind, and I'm more than happy to help. Now, the last things I want to mention, we're going to go into the breakdown that's occurring. So when you think of the process of cellular respiration, you're thinking of the metabolic process. You're breaking down a glucose molecule, and if you break things down, what is that called? Well, remember I said, picture a cat scratching apart a sofa, just violently scratching it apart. That's catabolism, or catabolic reaction. Okay, so catabolic means breaking something down. So cellular respiration, I could ask, is cellular respiration catabolic, or is it building something up, which is anabolic, like Arnold Schwarzenegger building up all those muscles, anabolic. So if you've heard anabolic steroids. Okay, so remember we said catabolic, cat scratching apart a sofa, breaking it down, is metabolic reactions in which something is broken down. Anabolic, like Arnold Schwarzenegger and steroids, is building up. So once you remind yourself of the difference between those terms, then you can ask yourself, is cellular respiration, when glucose is being broken down, catabolic or anabolic? Well, it's catabolic, okay? So that's a question that I can ask you. Now, catabolic reactions or catabolism, when you break down molecules, you are releasing energy, okay? That ATP produced by catabolism drives anabolism, okay? Meaning catabolic reactions, breaking down molecules, releasing ATP, drives anabolic reactions, which means building up molecules, okay? Because it takes energy to build something. Think of how exhausted you would be if you had to build a wall, right? It takes a lot of energy. If you had to build a house, a lot of energy. Build a car, any building, that's going to take a lot of energy, okay? So with that, you are then consuming energy that had been gained through catabolic reactions. Okay, so if you think about it, broken down or catabolized intermediates from the respiratory pathway, the cell respiration pathway, can then be used for different anabolic pathways to build up other molecules. Because remember, we said that in this cellular respiration, where you have the glucose being broken down into ATP, right? You got a lot of ATP being produced as the glucose was broken down into all of its intermediates, such as pyruvate. So you have a lot of ATP produced. That ATP could then go toward building other molecules, building the biomolecules, for instance, fats, carbs, proteins, lipids, okay? So when we talk about catabolism, anabolism, make sure you know the difference. So catabolic, breaking things down, picture that cat again, scratching up. Anabolic, building things up. And they're connected in that when you break things down in catabolic reactions, you are releasing ATP, energy, right? That then goes toward building things up in anabolic reactions. Again, if you have any questions or are unsure about this, just send me a message. And before we wrap up, I just want you to get one last look at the energy organelles, because in this current lesson, we focused on cellular respiration occurring in the mitochondria. But in the next chapter, we're going to focus on photosynthesis, and that occurring in the chloroplast organelle. And each of these processes are opposite of each other. Okay, so you may notice kind of differences between them. Okay, for instance, you notice in photosynthesis, you have carbon dioxide reduced to carbohydrate, but today in cellular respiration, we said carbohydrate is reduced to carbon dioxide. Okay, because these two processes are opposites. Okay, but again, this is just to remind you, cellular respiration, when you think of that, I want you to think of the mitochondria as the organelle. Next, in photosynthesis, when you think of that, I want you to think of the chloroplast as the organelle. And that is it for cellular respiration, okay? I know sometimes it's a lot at once, but again, a lot of it is just repetition to really solidify the concepts for you. Now, again, with any chapter that we finish, make sure you are now making this chapter as part of a study guide. Okay, again, focusing on any of the things I kept repeating, on bold, colorful terms, on questions that I asked throughout the lesson or had on the slides. Okay, that's always important. Terminology is important as well. And start to think if any of these concepts that we covered in this current chapter are things that you might want to ask a question in the discussion board post that you'll have to do by the end of the week. Okay, if you have any questions whatsoever, you want practice problems, or for me to further explain any of the concepts or put it into different words, then just send me a message in the Remind app as always. Okay, never hesitate to ask me any questions. Okay, thank you and have a wonderful day.